But the particular introduction today is we're going to start this series with Dr. Rafael Tapia Rojo. Rafa is a new appointed lecturer in biological physics at the Department of Physics at King's. And he's an expert on protein mechanics. So he should be able to tell you from the smallest sort of viewpoint, so from the molecular level and perhaps even from the atomistic level, how proteins and how different molecules in general behave under force. And he'll probably introduce you to the techniques, I don't know, the different nanomechanical techniques by which you can sort of perturb and sort of extend and recoil individual proteins, and how this has sort of importance in the broader scheme of biological programs or biological processes that occur at the cellular level, tissue level, organ level, and even beyond. So, so I just wanted to say hello to everyone, to introduce you to this part of the program, and in particular to introduce you to Rafa. So I'm sure you'll enjoy. So thank you, Rafa, for doing that.
Yeah, sure.
Thank you all for being here, and I hope to see you guys very soon. I think as soon as next week, where we're going to have the formal launch of the program. So I'm going to be able to sort of welcome you in person very soon. So thank you all, and I'll see you later. Thanks, Rafa.
Many thanks, Sergi, for the introduction. And yeah, Sergi explained the idea of this talk. I will talk a bit about the field in which I work, which is a field of single molecule for spectroscopy, and in particular focusing on one particular problem, one broad set of problems that you can study with these techniques, which is how proteins behave under force. So single molecule for spectroscopy techniques are widespread in biophysics for addressing many different questions. And in particular, today I will focus here on protein dynamics, which is what I work on. So also, before starting, if you want to stop me in any moment because you have questions or whatever, I think it's best that you do that instead of like waiting to the end, so that way I can more or less know how are you following everything I'm saying, and so just interrupt me at any moment if you want. So the way that I've organized this class is in this kind of three general sections, okay, where I will first overview a bit. I will try to motivate a bit why it is important. It is an interesting scientific question to study protein dynamics under force. I will try to motivate, let's say, the broad scientific question. Then I will move a bit to the experimental techniques that we have to study protein dynamics under force, which are namely three main techniques: the atomic force microscope, or the AFM, optical tweezers, and magnetic tweezers. And I will talk briefly about the different differences, advantages, and disadvantages of each of the techniques, depending on the kind of problem that you want to address. And finally, I will talk a bit about the, let's say, an analysis framework, the theoretical framework that we have, the kind of tools we have to interpret these experiments, basically what kind of data you get and what kind of tools can you use to extract information or to learn something from these sort of experiments. Okay, so I'll start with the first section, which is why do we want to study protein dynamics under force? And here I will just make a brief overview and present like three practical examples where I believe are really interesting and where force spectroscopy actually had a lot to say, and also probably still there are open questions in these three problems. So being this mechanobiology program, probably is not really necessary to motivate why mechanical forces are important in nature and why mechanical forces are important in biology, because we know that across all scales, from the organisms to organs or to single cells, mechanical forces play an important role in many biological functions. What I want to emphasize here, which is the system that I'm going to be speaking about, is that under all this complexity, under all these complex mechanical processes, what underlies is single proteins that bear forces, single proteins that are under force and perform their functions under force. So thus, if we want to understand all these complex processes, such as, for example, heartbeat, muscle contraction, adhesion, cellular motility, all this kind of example, in the end, what we have is some protein, specialized proteins that have evolved to develop their function under mechanical forces. And actually, by correlating their dynamics under force with their function, we will be able to understand these processes from a molecular perspective. Okay, so here I have three examples that I think have contributed a lot in the field of force spectroscopy. Starting with muscle contraction and the study of the titin protein, which is one of the very first systems that were studied with the use of force spectroscopy techniques. So obviously muscles and force, it's a very obvious relation. So every time we contract a muscle or we stretch a muscle, all the tissues, all the cells, all the proteins in these tissues are subject to cycles of stretching and contraction. And here at the smallest level, at the smallest module that forms a muscle, which is a sarcomere, we have three main filaments, which is actin, myosin, and titin, which is the protein I'll be speaking briefly about today. So titin is the largest protein in your body. It's a megadalton scale protein that is a single polypeptide chain arranged in many different domains, like bits on a string, so to speak, that is anchored between here the myosin and the set disc. So obviously it is under stress. So every time we stretch a muscle, titin stretches, and every time we contract a muscle, titin contracts. So thanks to the use of force spectroscopy techniques over the last 20 or 30 years, we have starting to understand how these titin domains behave when you stretch them, when you apply mechanical forces. For example, we now know that when you take one of these individual modules and apply a mechanical force, you unfold them, you stretch them, and you can trigger a conformational change, which is the unfolding of this protein domain. But even when you decrease force, this conformational change is reversible, and actually the protein can refold under force and acquire again its folded conformation. So interestingly, also the different domains along the titin I band have different mechanical stability. So there is a hierarchy of mechanical stabilities. This means that the domains that are in the proximal regions close to the set disc are weaker. They need lower forces to unfold. They can bear lower forces, while as we move on to the distal region, these domains are stronger and stronger. They can withstand, let's say, higher forces. So obviously this is something really interesting that I believe is related to the function of titin in the muscle, which we still do not understand very well, how it contributes to muscle elasticity, of perhaps if these conformational transitions that it can undergo under force, these folding and refolding transitions, might have some contribution to the function of muscles, to muscle contraction. So a totally different system, a totally different example is that of bacterial adhesion, which is, for me, it's quite a striking example, because every time a bacteria tries to infect tissues, it attaches to a host cell, and it does so using some kind of hair-like structures, which are termed the pili. And these pili are protein structure made by the concatenation of different protein domains. Several hundreds of protein domains concatenated one after the other, capped by a tip protein, which is an adhesin that establishes the chemical anchor between the bacteria and the host cell. Now, if you think about it, these bacteria are subject to huge mechanical challenges, to a lot of mechanical stress. You can think, for example, the mucus flowing, or every time we cough, every time we brush our teeth, we're basically applying mechanical shear stress to these bacteria, trying to detach them to prevent the infection. And interestingly, what we are now learning, thanks also to the use of these kind of force spectroscopy techniques, is that bacteria have evolved really smart chemical strategy that confer the proteins that form their pilus with outstanding mechanical properties. For example, many bacteria have isopeptide bonds, which is a kind of covalent bond that form between side chains of amino acids. And basically these isopeptide bonds can clamp the N and C terminal of the protein and render an extensible protein, so that when you try to pull on them, when you try to stretch them, these proteins won't unfold, won't change conformation, and they will remain folded for, no matter how hard you pull. And actually this is really relevant for their function, because if you can think, if you had like really soft proteins that will unfold at really low mechanical perturbations in this context, they will be basically quickly cleaved by proteases that are, in our body. So obviously, if you're a bacteria and you want to remain attached, you want a really strong mechanical, a protein that is really strong from the mechanical perspective that will allow you to resist all of these huge mechanical challenges. And also on the adhesion level, the chemical interaction between bacteria and the host cells is typically mediated by different kind of chemical strategies, typically that involve catch bonds. And catch bond is some kind of a slip knot in the sense that as you apply higher forces, the bond becomes stronger and stronger. And all these kind of properties that relate protein chemistry and mechanics can be directly studied with the use of force spectroscopy techniques, where we can learn the reactivity of these bonds, where we can learn the mechanical properties of this protein, and perhaps even design some kind of strategies to interfere with this kind of chemical strategies that perhaps in the future could open the gate to, let's say, have some kind of rational therapy strategy that targets directly the mechanical properties of these proteins that basically establish the anchor between a bacteria and its substrate. But yeah, and even in a more general way, most cells in our body need to sense forces and detect forces from their environment, either from the extracellular matrix or from neighbor cells. And actually, these forces and these force sensing properties are really important for many cellular processes, such as cell spreading, migration, differentiation, etc. There are a plethora of processes that are guided by the ability of cells to interact with their mechanical environment. And to this aim, cells are equipped with different specialized proteins that are able to sense and respond to these forces in a particular way, and basically translate between the language of forces to the language of biology, which is typically biochemistry. Basically, this is what is known as mechanotransaction, is the ability of cells to detect forces and convert these forces into into signaling pathways that eventually can develop into gene transcription programs that allow the cell to dynamically respond and adapt to this mechanical environment. So here, and very different from the other two previous examples, the one Titin or the bacteria pili, the force sensors the protein force sensors that cells have are typically exposed to really low forces, like very, very low forces, while we are talking about forces of hundreds of piconewton. In the previous cases here, we are talking about forces of just a few piconewton, which are the kind of forces the protein chains inside your cells experience. And this is a challenge from a technological and an instrumental point of view, because it's not that easy to study proteins under such low force conditions. So in order to understand all these mechanosensing and mechanotransaction processes, we need to develop new technology that allows us to study the dynamics of these proteins under the kind of forces they experience in the cells. And this is something that now we are doing. For example, here I show you, I will explain better later what this means, actually, this kind of recording. But basically, this is the dynamics, the end-to-end extension of the tailing protein measured over long timescales of the kind of forces that it experiences in the cell, that typically around 5 to 10 or to 15 piconewtons. So now with these new technology tools, in particular, this is magnetic tweezers, which I will explain later, we are able to measure and understand how these proteins behave in the kind of mechanical conditions that they experience in the cell. And not even that, but also now we are starting to be able to understand how these proteins interact under force, because protein interactions under force are critical for these mechanotransaction processes. And here, one of the classical examples for me, a key mechanism is that of cryptic binding, or even cryptic post-translational modifications, that basically mean that many of these proteins harbor binding sites that are cryptic, that are buried inside the structure of the protein, the fold of the protein, so that without force, these binding sites are hidden. But when you have force, this protein can undergo a conformational change, such as unfolding, expose this binding site, and allow a new binder to basically recognize this site and bind. The classical example of this, which was described in this paper in 2009, is that of tailin and vinculin binding. Basically, tailin and its main partner vinculin. Tailin has about 11 different binding sites for vinculin across It's different domains, and they all are cryptic, meaning that you need to unfold, you need to unravel this binding site mechanically, expose them to vinculating for it to bind. And now with these new techniques, we're being able to actually measure in a really precise way how these interactions occur under force and how forces regulate these interactions either for binding but also for unbinding. And now something that I want, before going into instrumentation and all that, something that I want to stress, and I mean, there's something I want that you have very clear, is that proteins under force behave in a really different way than proteins inside a test tube. So for example, protein folding and protein unfolding is really different when it occurs under force or when it occurs by temperature or by some chemical denaturants, which is the kind of typical classical protein folding experiments that you do in a test tube using like classical biochemical assays. So when a protein is stretched, when a protein is it's under force, it unfolds and it extends linearly along the end-to-end length, unraveling a large extension that was previously trapped on its folded conformation. So in order to study these kind of processes, which are the kind of processes that are occurring at the cellular level, we need to use different techniques. We cannot rely on the classical bulk like biochemistry or structural techniques that allow us to study ensembles of molecules in solution. We need to design and we need to implement new instrumental techniques that allow us first to tether a single protein, apply force to it, and measure the conformational changes as a function of these forces. Okay. So now that I believe I've motivated a bit why we really want to study, why we really want to measure protein dynamics under force, I'm going to introduce different techniques that you can use to this aim. And I will basically be overviewing the three main techniques that exist, which are atomic force microscope, optical tweezers, and magnetic tweezers. And again, the idea behind all these three techniques is quite similar, because in the end, what you want to do to study protein dynamics under force is first to apply force, and second, to measure conformational dynamics of this protein, typically through the end-to-end changes. And all these techniques, what they try to do is to tether a single protein between some substrate and a force probe so that you can stretch it and at the same time measure its extension changes. But it's one of these three techniques, use a different approach for doing this. So the AFM, which is this technique depicted here, uses a very small cantilever, which you can think as like a nano fishing rod, so to speak, that fishes a molecule so that you can stretch it, and by measuring how this cantilever deflects, as I will show you later, you can measure force. Optical tweezers uses light to trap small bits, so that when you have a molecule tethered between two bits, by manipulating this optical trap, you can apply stretching forces to your molecule. And magnetic tweezers uses a magnetic field that you can generate with a simple pair of permanent magnets to apply forces to a superparamagnetic bit, so that by having a molecule tethered to that superparamagnetic bit, you can stretch it. So I'll start explaining in a bit more of detail the atomic force microscope. So atomic force microscope are widely spread in many other areas of science, are widely used in many other areas of science. But here I'm going to focus on describing how you can use atomic force microscopy for measuring protein dynamics. So simply, and the typical arrangement what you have when you use an AFM is this kind of polyprotein arrangement, where you have similar to protein structure that you have actually, like in the case of titin, peelings, etc., where you have different domains arranged one after the other like beads on a string. So typically you can engineer, when you want to study a protein, a polyprotein chain made of different domains repeated, all the same or different, along the same, let's say, polypeptide chain, so that you can anchor that construct to a gold surface, to a substrate, and to a cantilever tip, which again have the idea of like a nano fishing rod, like a micron-sized fishing rod. One of the ends of the protein is anchored to the substrate, which is covered in gold with a terminal cysteine that allows to have a covalent anchoring, while on the other end, the interaction between the cantilever and the other end of the molecule is random. It's non-specific. It's literally like fishing. Again, so you will just press your cantilever in different positions along your gold surface until you successfully catch a molecule and you can stretch it. Okay, and then the gold surface is mounted on a piezoelectric actuator, and piezoelectric actuators are really common in any kind of these techniques because basically allow you to control position by controlling voltage. So it's a really easy way to control position. So simply by changing the voltage that you apply to this piezoelectric actuator, you can approach the surface, or retract it. And by retracting it, you can apply increasing forces and observe like these conformational changes on this molecule, that as you increase force, the molecules will unfold and the whole protein change will extend under force. So here something that is important is how you measure forces, and the typical mode that you call here on this kind of experiments is the force extension mode, because basically you retract your surface, or you extend, you control this distance. And now, as for example, a protein unfolds, it releases some tension. So the deflection of this cantilever, which acts basically as a linear spring, will change. So that by measuring this deflection, which you have calibrated, you can basically measure the force that your protein is exposed to. And the kind of recordings that you obtain is this classic and beautiful sawtooth patterns, which is what they are called. When you have, for example, an octamer of some protein, which is 8 identical repeats of the same protein. As you increase force, this is extension, this is force, you obtain this classic sawtooth pattern peaks, where each of these peak events mean one single domain unfolding at some particular force and basically liberating all this hidden sequence that it had on the fold. Now, another mode of operation of the AFM is what's called the Force Clamp Afm. So here again, on the regular AFM, you retract this gold surface, so you stretch your molecule and you measure force. However, sometimes what you want to actually is to control force and measure protein extension. So here on an AFM, what you can use is a PID feedback system, which is basically an electronic circuit and a standard electronic circuit that allows you to correct the position of this piezoelectric actuator so that the tension, the force that you're applying with your cantilever is always constant. And that way you can subject your molecules to stretching forces that are controlled. So here you measure extension as a function of time at different forces. So for example, now instead of having these sawtooth patterns that you had before, if you remember, you have this kind of staircase patterns, where each of the steps correspond to the unfolding event of one protein, so that your whole molecule gains extension at 100, for example, and 20 piconewtons in this case. Now, the advantage of force clamping, that's why many people like it a lot, for example, me, is that you can also monitor, you can also measure the opposite reaction, not just unfolding by applying increasing loads to your molecule, but also refolding by decreasing the force and then having a high force pulse again and seeing if your molecule was capable of acquiring its native conformation again, that you can map here by comparing the mechanical stabilities of the different pulses that you explore. Okay, but here, again, I stress that to do this, to have these force clamp conditions, you need an electronic feedback system that allows you to correct in real time the position of your piezoelectric actuator. Now, optical tweezers have a totally different principle of action, which basically uses light to trap, as I was saying, 2 beads. And well, this is how an optical tweezer setup looks. It's kind of a complicated optical setup, because what you want to do is to focus tightly in a tiny region 2 lasers, so that you create a region where you can use like a linear potential that you can use to trap objects, small objects, such as beads, but also you can use optical tweezers for trapping like cells, bacteria, whatever. And simply by trapping these beads, that if you have a molecule tethered between the beads, by controlling the position of the laser traps, you can apply stretching forces and again trigger some conformational changes to your molecule. Okay, here I'm highlighting this because this is different than the case of the AFM, where you were having a polyprotein, where all the, let's say, molecule that you had between your substrate and your cantilever was a protein of interest. Here, typically, you need to isolate the molecule of interest with long DNA handles that basically allow you to have the beads far apart. And this is because you're using light, so you can literally fry your molecule if these bits are too close. So you need basically to space all these. And for that, typically people use micron-sized DNA handles that create all these distance between the trap. And again, the kind of recordings that you get with optical tweezers is not that different from the AFM because the mode of action is the same. It's force extension. You control the distance between the traps and you measure force. Okay, so as you move away the traps, the force on the molecule, the tension on the molecule increases, and then you can have like this rupture event that indicates this is the release of tension because the protein unfolded. And then, you know, from these kind of recordings, you can extract some properties that I will explain later. Basically, the idea that I want to highlight here is that in contrast to the AFM, where we were applying forces of hundreds of piconewton, optical tweezers typically work better on a lower force range and are really good at applying low forces, but cannot like go above a certain value of forces. Okay. Now, just to finish this kind of instrumental review, I will talk about magnetic tweezers, which is the last technique, and I believe is the most straightforward one because it's based on a really simple principle, which is using magnets, same as the magnets that you have in the fridge in your kitchen, to apply forces in the piconewton range to molecules. So here, what you have is a single molecule tethered to a glass cover slide and to a superparamagnetic bead. And a superparamagnetic bead, superparamagnetic materials are you know, they can develop very high magnetic moments in the presence of magnetic fields. So basically they are attracted to magnetic fields. So simply by controlling, by calibrating a magnetic field that again, you can apply with permanent magnets, you subject your molecule to forces in the range of piconewtons. And simply by approaching these magnets, you will increase the force. And by retracting these magnets, you will decrease the force. As simple as that. Now to measure the extension of your molecule, you simply use a commercial microscope, a regular microscope that you use to track with simple video or image analysis techniques. You use to track the position of your bead when it's observed, like from below here. Simply what you do is you compare the pattern of the diffraction rings of your magnetic bead, which is the one that will be moving up and down, following the dynamics of your protein, in a reference bead that is tightly anchored to the glass cover light and has no magnetic properties, so basically it does not respond to force. Okay, so simply by comparing the diffraction rings of this bead that is moving and this bead that doesn't move, you're able to infer the extension of your molecule. So as your molecule goes up and down with force, because basically it's unfolding or refolding, it's undergoing some kind of conformational transition, you can measure extension as a function of time. And typically you obtain these classic hopping recordings where your protein is transitioning between a low extension state, which is the folded conformation, and a higher extension state, which is the unfolded conformation. And you can track the stochastic transition between these two states at different forces. And importantly, and this is something again I want to stress, magnetic tweezers are good at low forces, but also have intrinsic force clamp conditions. So different from the AFM or the optical tweezers, where if you wanted to work under force clamp, if you wanted to control force, you need an external feedback, an active feedback system to control force. Here, just by construction, just by the way the system, the instrument is designed, you control by default force and you measure extension. So you don't work on force extension mode, you work on force clamp by default. This is the way it works. So it's a really good technique if you want to study proteins under low forces during very, very long timescales and in equilibrium conditions. And this is also an advantage of magnetic tweezers, that is intrinsically more stable than the other previous two techniques. And again, I show you this trace of tailing dynamics measured over several hours at a low force of 8 piconewton, which is the kind of force it experiences in the cell. Okay, and even you can hold proteins for even several days, as this trace that I show you here with a different protein that has much slower dynamics. For example, here this protein transitions every 20 minutes or half an hour. So with techniques that are not able to explore such long time scale, you know, basically this transition would be invisible. And here with magnetic tweezers, you're actually able to resolve all these low processes that many times are actually relevant in physiology. So just to finish, this section. Here I have like a little comparison of the three techniques so that more or less you have an idea, like a sort of take-home message of why, I mean, for what its technique is good and for what it is not. So in terms of time resolution and stability, the AFM and the optical tweezers are quite similar. You have a really high time resolution. You can really resolve very short events, even on the microsecond time scale, because basically you're using light, you're using laser to monitor, to sample your changes, so you can do that at really high rates. While magnetic tweezer that uses simple video technique, like simple image analysis techniques, just can resolve up to a few milliseconds, just in the millisecond time scale. And by contrast, while AFM and optical tweezer have a limited stability, you can just measure a single protein for a few hundreds of seconds or perhaps a few minutes. In magnetic tweezers, you can typically extend these measurements for very long times, up to even, as I was showing you before, several hours or even days. And in terms of forces that you can apply, optical tweezers and magnetic tweezers are quite similar. They're really good at low forces, while the AFM excels at applying high forces. So again, depending on the kind of system you could be interested in, there's, AFM is really good for for proteins that are mechanically very stiff. For example, peeling proteins, tighten proteins, they typically respond to forces on this range, on the 100 to 100 piconewton range. While if you want to measure, for example, proteins that work inside the cell as force sensor, you probably want to go with optical tweezers or magnetic tweezers because they're really good at measuring proteins in this time scale, in this force scale that is relevant, for example, for mechanosensing proteins. And even more generally, another difference, again, I stress this, is that AFM and optical tweezers by default work in the force extension mode. So you control extension, you control the distance between your traps, and you measure force as a stochastic variable, basically the force that your protein is experiencing, while magnetic tweezers have passive force clamp conditions. So you control force and you measure extension. Okay. So now going to the last section of this class, I would like to overview a bit what kind of data you obtain with these kind of techniques, and what kind of analysis, what kind of, let's say, parameters are you looking for in order to learn something about your system. And here I will kind of divide these in two different mini sections, one related to protein elasticity. So you'll see now basically is the relationship between unfolded proteins and force, how the extension of that protein scales with force. And the other one is mechanical stability, like how you can relate unfolding forces or the forces that with some conformational changes occur with the properties of that protein. So starting with the properties of protein elasticity, this is basically how proteins extend under force. And here I want just to stress concept, which is that unfolded proteins are unstructured polymers. So basically they behave on unstructured polymers. That's random polymers with no structure that they stretch under force. And you can look here at this little animation, which is a molecular dynamic simulation of a specific mechanosensing domain, allowing protein that under force undergoes this kind of transition. So this is the kind of things that is happening when you do a force a mechanical experiment. I will plot the video again. So basically, as you apply force, the molecule loses its structure, its native structure, and extends under force to some particular extension. And basically, the relationship between this protein extension and force is what basically connects or basically determines the elasticity of these proteins. And here, the idea is that proteins, unfolded proteins, obey the laws of polymer physics. Okay, so they are polymers. And this is different from the case of hook and spring, which is the typical elasticity model that we are used to. Okay, the kind of high school physics system where you have a linear relationship between force and extension. And the quantity that relates both these quantities is the stiffness. So if you have a very stiff spring, you need a lot of force to stretch it. And if you have a soft spring, you don't need that much force to stretch it. And the relationship between force and extension in hook and springs, in linear springs, it's a line, it's a linear thing. So this is not the case in proteins. So the rules of polymer physics basically are the rules of entropy. So when you have an unstructured polymer in the absence of force, it collapses to a configuration, kind of a globular configuration that tries to maximize its entropy. So it tries to have as many degrees of freedom as possible. As you stretch, as you apply force, basically what you're doing is removing these degrees of freedom, removing these available configurations and reducing the entropy of your system. So simply protein elasticity, you can think about it as a tug of war between force and entropy. Entropy tries to collapse your protein to maximize the number of available configurations while force is trying to stretch. And this gives rise to this kind of nonlinear behavior that you already can see in the sawtooth patterns, as I'm showing you here, where the stiffness is not a constant, it's not a line, the slope is not a line, but it's a nonlinear quantity. At the beginning, when you start pulling on an unstructured polymer, here, as you can see, is really soft. The slope is really small, and you gain a lot of extension with very little force. But now, as you keep on increasing the force, the protein becomes stiffer and stiffer, and it gets harder and harder to extend it. Okay, so this is the kind of behavior that the protein under force has. At the beginning, with low forces, it's really soft, but as you keep on pulling, it becomes stiffer and stiffer, and it's harder and harder to extend it further. And to explain these properties, you can basically use simple polymer physics models, such as the Worthington model or the Free-Untitled model that I will explain on the next slide. So I'm not going to get into the math or the physics of this model, but just give you kind of an intuitive idea. So the Worthington model has this expression, which, as you can see, is it's not similar, it's analogous to the Hookean spring, but much more complicated. Well, here it was this simple linear expression. The Worthington model is much more complicated expression that gives rise to this nonlinear elasticity that proteins have. And here you have two parameters that are key and basically characterize the elastic properties of proteins, which is the contour length and the persistent length. So the contour length is a really straightforward parameter to understand. Basically, it's the maximum extension a protein can have. Simply, is you have all the amino acid perfectly aligned along a line, and you just sum the distance. That is the contour length. It's the maximum extension a protein can have. Now here, and because of these entropic properties that I was telling you, a protein never reaches its contour length. You will need infinite force to really align everything, because that will be the state of zero entropy. Okay, so proteins approach to their contour length as you increase the pulling force, but they never reach it. And the second concept is persistent length, which is kind of related to stiffness, but in a different way. And for this, I think it's a good analogy to think of spaghetti. And if you think of raw spaghetti, they are systems with high persistent length. Basically, mine having a gigantic spaghetti that you hold, and you see what's the extension, what's the length of this spaghetti until it starts to bend. Okay, that's the persistent length. Basically, it's the length scale over which a polymer behaves as a rigid rod. Okay, so for example, a raw spaghetti will have a high persistent length because probably you can hold it for several centimeters until it starts bending. However, when you cook spaghetti, you massively decrease its persistent length because it now becomes like a floppy and soft entity. And obviously, if you would hold this gigantic spaghetti now cooked, it will bend really quickly. So that is a low persistent length. So persistent length basically tells you this: what's the length scale over which your polymer behaves as a rigid body. And here I'm plotting different Worthington models for different persistent lengths. Here, the raw spaghetti thing is the red thing, where over the very first few piconewtons, you extend a lot. It's really soft, but then as you keep on increasing force, you don't gain that much extension. So it's a really nonlinear shape like this. While if you have a polymer with a lower persistent length, such as in this case, basically it has a much shallower force dependence. But the idea is the same, that over the very first few piconewtons, you acquire most of the extension, available extension of the system, and then as you keep on increasing the force, it becomes harder and harder to extend it. And while it approaches to the contour length, it never really reaches it, never touches it. Okay. And now the other model of polymer elasticity is the freely jointed chain model. Okay, which is it's kind of completely analogous. It's the kind of description it does is exactly the same, but the expression is a bit different because it doesn't relate force with extension. Here you can see that Worthington model, it will basically tell you what's the force you need to put a polymer to a particular extension. Here, the jointed chain tells you what's the extension of a polymer for a particular length, but the idea behind it is exactly the same. Here you have the contour length, same parameters, same magnitude as before, And here you have the Kuhn length that plays the role of persistent length, and actually they are related. And you can think that the Kuhn length of a polymer, it's roughly twice the persistent length of that polymer. So when you plot the free-ended chain model, simply it's like the same curve that I had on the previous slide, but like flipped like on a mirror. So you have extension on the y-axis and force, but the idea is exactly the same. Systems with very high persistent lengths, like the red curve, stretch a lot over the very first few piconewtons, while systems with lower persistent length, they have shallower force responses. And if you think on biomolecules, the red curve will be DNA. DNA is it's a molecule with a really high persistent length, so it's really stiff. Over very low forces, it stretches a lot, while proteins more or less behave like the black curve. They have persistent lengths of about 1 nanometer. All right. Now I'm going to, just to end, to talk about a bit of what kind of properties you can extract from the force response, from the force-dependent dynamics of proteins. And I will divide this again on the two different modes of operation that you can have in force spectroscopy, which is force extension and force clamp, starting with force extension. So force extension, again, remember, you measure force as a function of the distance between your traps. So you apply an increasing load to your molecule until it breaks, until it ruptures its structure, and it unveils all this hidden length because it's unfolded. And then again, you go to the next molecule, and that gives rise to these characteristic sawtooth patterns. So it's important to know that sometimes you have molecules with very low mechanical stability, so these peaks will be low, and sometimes you have molecules with high mechanical stability, so these peaks will be very high. And when you have a protein, a polyprotein made up of different domains, some with low mechanical stabilities or lower mechanical stabilities, such as the I-27 here, and some with higher mechanical stabilities, such as the I-28. What you have always is a hierarchy in the mechanical stability. So always weak things unfold first and stronger things unfold later, regardless of what's the sequence across your chain. Also, something important that I kind of forgot to tell you probably at the beginning is that everything we measured at a single molecule level is stochastic. This means that every protein, every time you interrogate it, behaves in a slightly different way. So what you measure, what you recover are distributions of some molecular properties. That is what you can analyze and what you can use to understand your system. So that's why, for example, you can see that although all these peaks are roughly of the same height, they are all slightly different. Some proteins unfold at a bit higher force, some proteins unfold at a bit slower force, but overall you can reconstruct the distributions of these unfolding forces in this case and learn something about, for example, the mechanical stability of the protein. If you take like this simple landscape model, where you would consider that your folded protein is kind of is your system here on this energy well, unfolding is basically overcoming a free energy barrier, okay, that at low forces is very high, so you won't unfold. But as you keep on extending, as you keep on separating, for example, your traps in optical tweezers, what you do is decrease this barrier. So as you keep on pulling, this barrier is lower and lower, and at some point it's favorable. Stochastically, you can overcome this barrier, and by overcoming this barrier, you unfold, you increase. So that's the kind of model that typically you use to understand these processes. So there are many different theories that try to basically relate the distribution of unfolding forces with some molecular properties and some structural properties of the protein that you're using. The simplest of them all, and probably the one that is used more widely, is the Bell model. That is the simplest of them all, that basically tells you that the average of the rupture force is related with, it depends logarithmic on the pulling velocity. So the idea here is that if you pull at really low velocities of your system, you can think that it's equilibrated in every moment. It will unfold at the lowest possible force, right, which is a equilibrium force for unfolding. While if you pull really, really, really fast, what you can think is that you're not equilibrating the system, so you would need higher forces to actually observe this unfolding event. It's like thinking that your molecule needs a bit of time to realize how high the force it is to unfold. And that's why with the pulling velocity, the rupture force increases. But anyway, if you measure rupture forces at different pulling velocities and you feed that to the Bell model, basically you can obtain some information about your system that namely is this structural parameter, which people typically call it the distance to transition state, that assuming this simple model is basically the distance between your energy well that tells you the extension of the folded state and the top of the energy barrier. And also the other parameter that you will obtain typically is the intrinsic rate coefficient, which you can think about it more or less as the rate at zero force, roughly. But now, when you use force clamp, which remember is a completely different mode of operation because you control force and you observe extension, you can still recover the same properties that I was explaining you before, also using the Bell model on a different state. So here, the idea is that if you subject your protein to a fairly low force, for example, 25 piconewton, you'll have this staircase where it's unfolding event. All of these steps correspond to unfolding one particular domain, but as you increase the force, these unfolding events will become faster. Basically, you can think that the unfolding time gets shorter as you increase force. And the idea here, again, with this kind of landscape picture is the same. As you increase force, you lower the barrier, so the chance of unfolding is higher. So you observe you have a higher rate, or a lower unfolding time, okay, with force. And here, Bell model, what predicts is that the rate of unfolding of a protein as a function of force has an exponential shape. So if you measure distributions, for example, of unfolding time, which they have in general an exponential distribution, they're exponentially distributed because it's basically a Poisson process that occurs with a particular rate, and you measure these rates as a function of force, you can fit that to Bell model, which is in semi-logarithmic scale, a straight line, and from the slope here, you can recover the same kind of information that you had in the force extension mode, which is the distance to transition state and the intrinsic rate coefficient. But again, as I tried to motivate before, I think one of the main advantages of force clamp is that you can change force, and if you are able to control low forces, you are not only able to measure and observe unfolding, but you are also able to measure and observe refolding, which is the opposite reaction. And actually, this gives you a much richer amount of information. So you can think here that instead of having this kind of one-way landscape where you overcome the barrier and basically the process is over, it's irreversible, you have more like two states, one energy well that represents the folded state and one energy state that represents the unfolded state. So as you increase force, what you're doing is tilting this landscape towards the unfolded state. So it's at higher forces, the unfolded state is more favored compared to the folded state. But in some forces, in some intermediate value forces, both states can be equally likely. So what you can observe is dynamics of unfolding and dynamics of folding. And this is kind of what I'm showing you here in this trace. We also obtain in magnetic tweezers for a polyprotein of protein L, which is a model protein that we use sometimes, where first at 40 piconewtons, you can unfold the full proteins and you can measure again all these unfolding rates and retrieve all this information. But now when you go to lower forces, you can see that you have the opposite process where you have steps that go, let's say, down, and each of these steps correspond to a single domain, protein domain refolding, but because you're in a range where this coexistence of transitions exists, you can have steps up and a step down, and you can have like this beautiful dynamics of folding and unfolding. So overall, you can reconstruct the rates both for unfolding and both for refolding that have the opposite force dependence. So refolding rate decreases really steeply with force in an exponential way as well. And even, I'm not going to stop, just give you this idea like really quickly, that now with new techniques that we are increasing the resolution, we are being able not just to observe transitions, but also to observe how, these transitions with millisecond or even microsecond timescales, and basically map and sample the excursions of the protein over its free energy landscape. And this gives you really rich information because it allows you to reconstruct the free energy landscape of your protein that basically contains all the information and codes all the response of that protein as a function of force. And by being able to basically map this energy landscape, we simply have all the information we need, all the information we want about how proteins respond to force. So now, just to finish, I'm just gonna very briefly outline some few conclusions about my class, on kind of take-home messages that I would like to have you clear. First, and the why, I hope I kind of motivated why it is important to study proteins under force. And the reason is that it's because many proteins in your body actually are under mechanical forces. And because protein folding under force is really different from the same reactions occurring on a test tube, we need new tools. We need new experimental techniques that allow us to actually subject these proteins to force and measure the dynamics, because that's the actual physiological conditions that they experience inside the cell. In terms of techniques, we have three main techniques, which are the AFM, optical tweezers, and magnetic tweezers. And again, it have different advantages and disadvantages. So basically, the idea is that depending on the kind of question you want to address, you have to choose a different technique. So if you want to study proteins with very high mechanical stability, you definitely want to go for the AFM. While if you want to study proteins with low mechanical stability, you definitely want to go to either optical tweezers or magnetic tweezers. And if you want to measure protein dynamics for very long times, then magnetic tweezers is your technique of choice for sure. And in terms of theory, there's much more obviously, but basically there are two simple ideas here of information of the kind of information that you can get from single molecule for spectroscopy experiments. First, elasticity of proteins, which is based on polymer physics. Again, they do not behave as linear Hookean springs, but rather as nonlinear polymers that are ruled by entropy. And also that based on stochastic physics, we can obtain a lot of information from the way these proteins respond under force that eventually can even allow us to reconstruct underlying landscape that basically contain all the information about protein dynamics. So that's all. So many thanks for listening. If you have any questions, you can ask now or reach me later through my email. So yeah, that's all. Many thanks for listening. Yes, okay, let me stop sharing my screen. Yeah, so I see some questions, so go ahead. Hello?
Hi, Rafael.
Hi. Yes, I can hear you.
How are you?
All good, thank you. How are you?
Yes, I'm good. Thank you for the talk. It was very interesting. Quite different to some of the stuff that I've seen recently. I have a couple sort of like kind of practical questions. So,
okay.
In terms of so we saw a couple techniques that use sort of DNA hooks to grab the protein as it was being pulled. How presumably the DNA is of some sort of known. It has some known structure, right? So when we're pulling, we know that the DNA, we know the DNA itself is not responsible for any of the unwinding, right?
Correct.
And it would always be the same.
Yeah, let me share my screen again. I think I can more, probably it's easier to illustrate it. Let me go to that slide you're talking about. I mean, actually what you're, let's go to this slide, for example. Actually, what you're saying is, yeah, it's an issue for spectroscopy, which is basically how you can isolate the dynamics of your protein of interest.
Yeah.
Because obviously you're never observing the dynamics of your protein of interest. You always observe the convolution of these dynamics of interest plus the handles and even plus your pulling device. So this is something that is intrinsic.
Yeah.
Here, if you look at this graph, the kind of forces that you explore are always below the BS transition, which is the winding transition of DNA that occurs at 65 piconewton. So that's why if you use DNA handles, you need to use proteins with low mechanical stability so that you can kind of uncouple the dynamics of your DNA handles from those of your protein. Also, with all these polymer things I was explaining, DNA handles, because they have a really high persistent length, they behave as very stiff handles. So basically, they have really little force response. Okay, because over perhaps the first piconewton, where your protein doesn't even knows that it's being pulled, it reaches almost its contour length. So on the regime of interest, they will behave basically as rigid handles.
Okay.
So that's basically the idea of why it's not bad. I mean, it's a good idea to study proteins in this shape. Still, there are also like many, discussion in the field about how these handles can influence, and people have developed different techniques to kind of deconvolve your raw recordings and remove the effect of handles. So yeah, that's an active, let's say, issue in the field, definitely.
Okay, I have one more sort of related question before I ask something maybe more interesting. Presumably you also have to isolate from sort of external forces, I guess, like sort of vibrations in the environment, and because obviously the forces are so small, something like I don't know, somebody walking by the machine, by
yeah. No,
how do you isolate it in that way?
It depends a lot on the technique.
Yeah,
I have to say. So typically all these machines, all these setups, you mount them on isolation optical tables, okay, so that they absorb vibrations and the influence of any kind of, as you say, any kind of, let's say, external perturbation is minimal, okay. Still, you can have, like effects on your recordings if someone enters a room or whatever. In the case, for example, of magnetic tweezer, which is the technique I use the most, it's really stable because basically with this idea of tracking simultaneously a bit where you have a molecule and a bit that is just anchored, okay, you basically you subtract any local effect, because whatever happens to one bit will happen to other bit, and because you're just measuring relative positions between both bits, you're able to basically don't have any influence of any of these kind of problems, simply because both bits will be equally affected to that perturbation, and that way it's why it is more stable. But again, all these techniques are mounted on optical tables to absorb vibrations and so on. And that is really, really important, of course. Yeah.
Okay. All right. Last one, so I don't take up too
no
much
problem.
time. But so you spoke about cryptic binding at the beginning. That's something that's particularly interesting to me, possibly relevant for my PhD. I was just wondering, are there any techniques where you are able to maybe observe the binding or analyze the binding under force?
Yeah. So here, probably, let me go again to the presentation, and I think probably I can answer better with this. Yeah, so here. So one approach, it's kind of the indirect approach, okay? Which is you measure your substrate, the dynamics of your substrate, in this case, tailing, okay? And then binding reveals as some kind of change in the dynamics. In this sense, if you can think of a protein unfolded and then something binding to it, you won't be able to come back to its native conformation, because basically there's like this big molecule bound to the stretch. So that's what we are seeing here, okay? You can see, for example, here, this is a single vinculin binding event that basically stops these dynamics, okay? And that's why the protein doesn't fold and refold anymore. So that is an indirect way of seeing this binding event.
Right.
If you want to really see it with your eyes, then there are different techniques that combine force spectroscopy with single molecule fluorescence.
Yeah.
Okay, so that you can have two recordings at the same time. One of the dynamics of your protein of interest, okay, of your substrate, same as here, and then also the fluorescence signal, for example, of a fluorescently tagged ligand, so that in the moment it binds, you will have, an increase of your fluorescence signal, and you will be able to directly see like these binding events. And there are many combinations of magnetic tweezers, AFM, or optical tweezers that combine force with light, and that way you have, two sources of information: dynamics and also binding events, and you can correlate them. So yeah.
Okay.
It's
Brilliant.
a really
Thank you very much.
Yeah, thanks for the questions. Anyone else, or yeah,
I have a question. So at the beginning, you stressed that the naturalization that happens when it's based on the typical tube approaches, like with chemicals or with temperature, is different than that one that happens when you apply forces, because when you apply forces and stop applying them, then the proteins recover their natural conformation. But if you are able to stretch the protein to an extent in which you turn it into a linear polymer, why do you think that how can this difference be happening? Because if you are stretching the protein and it forms a linear polymer, you must be breaking, for instance, the dithiol bindings between cysteines and so on. So it should be the same naturalization, you know. So why do you think that this happens? That
no, I mean,
it's different.
So here there are like two different things. I mean, what I tried. I mean, so first with chemical denaturation and all that, you can also recover your folding state. So you can also like go between folded and folded, and then by removing the denaturant, you can get back to the original thing. Same as with force. What I tried to stress is that these processes, these unfolding or folding processes under force as a denaturant or some chemistry, are really different. And the reason is, if you think, if you think it's because, if you think in reaction coordinates, it's because the reaction coordinate that is defined by force does not occur in the absence of force. So you're basically forcing your system to be stretched along a very particular coordinate or direction. So that's why it's a very different process. In terms of what you were saying of bonds and all, typically you don't break covalent bonds with force spectroscopy. Okay, you never break them because you need forces in the range of thousands of piconewtons, of nanonewtons to actually break bonds. Okay, so you simply break the non-covalent interactions that maintain the protein fold. If you have, for example, the sulfide bridges on your molecule, they will clamp, they will limit the extensibility of this protein. And actually, something that I then got into for spectroscopy techniques are really good for taking a look at chemical modifications at the single bond level. Because, for example, from the protein extensibility, you can infer if you have the sulfide bond, or you can put some chemical that cleaves the sulfide bonds and observe in real time how this reaction occurs and how it depends on force, etc. So basically, the idea is that you won't break covalent bonds with force in this range. Okay, and that's the reason why, they are able to recover its structure back. So did I answer your question more or less? Or?
Yeah, thank you.
Okay.